afternoon to everybody. And the recording is in progress. And I would like to welcome everybody to this last session. I'm not talking about the MCQ exam, but as a normal real life sessions, that will be the last one that we are going to encounter today. Really excited to watch a few more presentations. 4 groups will be presenting, group number 9, 10, 11, and 12. And that will be all about specific examples of biosimilars. So it will build on the discussions that we had over the last 2 weeks. I'm glad that a couple of some of the faculty are joining us. That is Stephanie and Peter Stonier, Maps, Unati, William, Leander Fontaine, and Birka Lehmann I've seen who have joined. Welcome to everybody. Welcome then also to ask questions. As usual, we will have one lead person for asking the questions from the field of biosimilars, and that will be Lise once the presentations are done. And the first presentation will be group number 9. Without further ado, let's get into this. And that is Cynthia Mufaveli and Prudence. And the topic is about Flixabi as an example for a biosimilar. Please take over and start with your presentation. Time-wise, as usual, it's about 10-15 minutes for the presentation, and the rest of the time is then available for discussion. And please make time for the discussion, because as the last 2 weeks have shown, the discussion is actually very interesting, and then helps also to understand a bit more the background. So it's also kind of a learning exercise and not just an exam. Good. Group number 9, please go ahead.
Thank you. My God, am I able to share this? Sorry, I'm just trying to share my screen. Just a moment.
We can see your screen, and if you could put that just in presentation mode, then that's it. Yeah, perfect. Yeah, we can see it in presentation mode. That's great.
Just a minute. Hello. Am I audible enough?
Yes, I can hear you, and we could see your presentation mode, but that has disappeared again.
Oh, really? Gosh. I think I'm having a challenge with my... Just a minute. Okay, so...
I will see your screen, and it's in presentation mode now. Maybe you click that hide button. Yeah, it works. So I think you can just talk around it. Yeah, so we can see what you show, but we cannot hear you.
Hello?
Yes,
Are you able to get me?
I hear you.
Is the screen being shared still?
Yes, the screen, but it is yeah, now it shows your presentation.
Okay, so am I audible?
Yes, now it is both. We can see and hear you.
Okay, thank you. So this is Cynthia Mwindachabe, and I'm representing Group 9 from Zamra. So this presentation was done by myself, Mufaweli, and Prudence Musonda. So ours was to look at biosimilars and their relation to biological reference products. So in the past weeks, we have had discussions on biosimilars. So I will not labor much to talk of what biosimilars are and biological reference products. So our task was to critically assess dossiers for the originator product, that is Remicade, and its biosimilar, which is Flixabi, as they relate to pharmacokinetics, human pharmacokinetics, clinical efficacy, and adverse events. So Remicade and Flixabi both contain the active substance infliximab. So infliximab is a medical product containing infliximab, which is an immunoglobulin monoclonal antibody. The reference product is Remicade, which has the same active substance, and was registered on the market in 1999. So Flixabi has been developed as a biosimilar medicinal product to the reference product Remicade. So the first task we had was to identify a specific indication such as rheumatoid arthritis and quote specific clinical trial data or evidence from the EPA during our assessment. So from the information we gathered, infliximab is indicated for rheumatoid arthritis, adult Crohn's disease, pediatric Crohn's disease, ulcerative colitis, pediatric ulcerative colitis, ankylosing spondylitis, psoriatic arthritis, and psoriasis. So for the indication rheumatoid arthritis, we noted that 2 clinical trials were submitted to start with. So the first one was a phase 1 clinical trial that was conducted in healthy German subjects. So this one was done to compare the PK, the safety, the tolerability, and immunogenicity of Flixabi and Remicade. However, since this assignment was for us to look at a clinical study that was going to support the indication such as rheumatoid arthritis, we picked on the second clinical trial study that was provided, which was clinical trial phase 3 that was conducted in rheumatoid arthritis subjects. So this one was also conducted in subjects from UK, Bulgaria, Poland, just to mention a few. It was done to check the safety, efficacy, immunogenicity, and PK parameters. So the main aim for this one was to just demonstrate the equivalence between Flixabi and Remicade. So to answer the first question, it was evidenced through the second trial, which I just mentioned, which is phase 3 clinical trial. It was evidenced through trial number SB2G31RA. This was a randomized double-blind parallel group multicenter clinical study to evaluate the efficacy, safety, PK, and immunogenicity of Flixabi compared to Remicade in subjects with moderate to severe RA despite methotrexate therapy. So apart from that, it was noted that no any other clinical trial was conducted to cover for the other indications that were put up in the product information for Flixabi. So for these ones, we noted to say there was extrapolation that was supported by TNF alpha binding assay and cell-based assay. That is for the rest of the other indications. So the other task was to reflect on how we would manage the licensing of the biosimilar in all indications that were licensed by the originator, Remicade. So we discussed to say licensing for the biosimilar Flixabi in all the other indications that have been licensed for the comparator will be based on the establishment of biosimilarity between the two products. Then it would be justifiable to extrapolate the equivalent clinical efficacy and comparable safety profile from the Flixabi study in rheumatoid arthritis patients to all other indications where Remicade has been approved. So from here, my colleague will take over.
Good afternoon, everyone. The next task, we were requested to consult the EPA and at the end to find an appendix that had a divergent opinion from some CHMP members. Then we were requested to summarize the arguments in a slide. So some members of the CHMP did not agree with the decision on recommending the grant of marketing authorization for Flixabi. And their argument was that biosimilarity to Remicade was not established due to the following reasons. They had said Flixabi appeared to be associated with a higher incidence of anti-drug antibody than the originator Remicade. In a phase 3 trial conducted in patients with rheumatoid arthritis, the efficacy of Flixabi was constantly estimated to be lower than that of Remicade. The efficacy was significantly lower in ADA-positive patients than in ADA-negative patients. Also, patients with rheumatoid arthritis investigated in the phase 3 trial were treated concomitantly with immunomodulator therapy and could have exhibited less immunogenicity than patients in other infliximab licensed indications. As a result, the consequences of the difference in ADA incidences and the impact on efficacy in these indications was unclear. So the proposal by the applicant to resolve the concerns related to immunogenicity in the post-marketing setting by initiating a prospective observational cohort study in the indications of ankylosing spondylitis and Crohn's disease is considered inadequate. Then in addition, it is questionable to what extent a non-randomized observational study can provide data that will effectively address the uncertainties. So the second task, we were requested to check in the EPA. EMA works with 2 rapporteurs from 2 different agencies who take responsibility for the entire process. So we were requested to note out the 2 rapporteurs from the EPA. So the rapporteurs, we noted that there was Jan Muller-Vegas from Paul Ehrlich Institute, German Federal Agency, and the co-rapporteur was Jens Hustenbeck from the Danish Medicines Agency. Then from these rapporteurs, Jens Hustenbeck was among the members that signed the divergent opinion. We were requested to discuss critically the arguments and form our own opinion from the EPA. And then what we noted was that the historical reported rates of ADA formation of Remicade have been around 8% in patients with rheumatoid arthritis treated concomitantly with methotrexate. The rates observed in both Remicade and Flixabi treated patients with the submitted RA trial were considerably higher. They had mentioned 5 to 6-fold, which was attributed to assay sensitivity in the current study. However, the applicant did not state the actual percentage of the ADAs recorded. And furthermore, due to the high incident rates of ADAs, efficacy was significantly reduced, which in our opinion is not desirable. In continuation, next slide. So as an increase in ADA increases loss of response and increase of adverse events, the risk therefore outweighs the benefits of having the drug on the market. And considering that for our country, we have other treatment options, a negative opinion would be given for this product. My colleague will take over from there.
Good afternoon. This is Mfaweli taking over from Prudence. The question that we asked, the other question we asked was, what is your opinion on voting in a regulatory process when it comes to efficacy and safety of a medicine? So our opinion is that it might cause bias on the decision made, and we strongly felt that voting was not a very good approach, and a decision should be based on sound scientific evidence and assessment of the risk and benefits that concerns of the particular application. Next slide. The next question was present a summary as to whether you would grant a positive opinion for your agency or not based on information available to you. So in this particular study, we noticed that the subjects did not include African population, and there were some issues on the ADAs, and based on that fact that there were issues on the ADAs as provided in the previous slides, and information was not available for the African population, we then came to a conclusion that the safety and efficacy in our ethnic group has not been ascertained, and absence of these studies, we thought that we would give a negative opinion as an authority, because we have an alternative on the market, which is Remicade. The next question. Summarize in one slide what you have learned from this case. What we've learned from this case is that firstly, voting is not a very good approach in making scientific and regulatory decisions, and there's some, since there are different views and some pending issues might have to be addressed at country level. Next slide. So this slide talks to the post authorization documents that we provided. So the applicant provided an update of the RMP. They submitted studies to be able to provide further information on the efficacy, safety, and immunogenicity, because initially there was a concern on the efficacy of the loss of efficacy due to the ADA development. So in the post authorization documents, this issue was actually addressed by the by the applicant, where they provided further studies, and they monitored the efficacy, safety, and immunogenicity. Next slide. So that's, as I stated, post-authorization. So the data was evaluated, and there was a 78-week extension phase data from the phase 3 pivotal clinical study, and from the company's safety database and real-world data from post-approval studies. So the results actually did demonstrate that the particular application demonstrated maintenance of the efficacy um, and safety. So the post-approval, actually, the applicant did address the issues that were there on the immunogenicity, safety, and efficacy. But I think our issue on this particular application is that when it comes to our ethnic group, there is no data that is available for this particular study. This ends our presentation. Thank you.
Yeah, thank you very much for that presentation, which was given by all the team members, which is excellent. And also for your own opinions. I think that's exactly what we want to raise the discussion and have also a look at your own, encourage you to have an own look at what other agencies have been doing, and then make your own decisions. And with this, I hand it over to Lise for more details in in-depth discussion.
Thank you very much. And thank you also to Group 9 for the nice presentation. And indeed, as Berend already appreciated, also for sharing your own reflection, your thoughts of how you would assess the dossier, how you would handle the specific case. That was much appreciated. I have first a few curiosities. In your presentation, you mentioned that you would more lean towards a negative opinion for this biosimilar candidate for several reasons, but you also mentioned that you saw less of a medical or patient need, let's say, to also have this biosimilar on your market, because you mentioned that you have a treatment alternative for Remicade. So I was just wondering if you could share with us if it's then another biosimilar, for example, or what this alternative treatment is that you have for Remicade in your country.
Sorry, I think if I'm getting you right, you were asking.
You mentioned
Okay,
a few times on the slide that you have a treatment alternative for the originator product, infliximab, on your market, and I was wondering what that product was.
That's Remicade.
Remicade itself. So you mean
Yes.
we have the originator, so we see less need to have a biosimilar alternative.
Yes, which we have. I think we're not satisfied with efficacy and safety in our ethnic group. They did submit data post-approval to demonstrate that there's no loss of, it's still safe and there's no loss of efficacy. That could arise due to the DAs, but in our particular setting, our ethnic group, they have not given us the information that we need. That's why we're saying that for us to be able to register this particular product, they should give us the information that we need, because we have an alternative on the market right now.
Okay, so meaning you have the originator, so you see less need for a biosimilar candidate to also be licensed on the market, especially because you have the originator. And do I understand correctly that for the originator, you also did not have ethnic data, or you did?
Sorry, kindly repeat the question.
So you mean, you also mentioned that for the biosimilar, one of the elements why you would not grant it a positive opinion would be that there was no testing in your ethnic population. Was this something that was required for the licensing of Remicade to have this additional testing in your ethnic population?
I think for it, maybe because it was registered way back and being the originator product, it might have not been a requirement for it at the time that it was being registered in our country.
So that can also be a reflection as a regulator to see, okay, what was submitted and assessed at the time that we evaluated and licensed the originator. And then one can also ask themselves the question, do we then want to have additional data or requirements for the biosimilar, as we have already quite a lot of experiences, multiple years of use of the originator in that population with supportive data of efficacy and safety and also that population. So this is also something that you could take into account. I was also wondering, because you nicely presented the divergent opinion that happened for Flixabi in 2016 within the centralized procedure at EMA. And I was wondering, it was at that time 2016. Do you think that today regulators might assess it differently, taking into account that we have already progressed some years and also taking into account what the changing attitude is of regulators towards comparative efficacy testing? Any thoughts on that? And also, of course, considering the fact that there was a lot of post approval data that then further contributed to the overall package of biosimilarity. Any considerations or reflections that you think that regulators might today take a different approach than what they did in 2016?
So I don't know if I got your question correct, but I think considering the fact that they provided us with more information, that there's been updates on what has post-marketing updates, I think um, regulators would actually consider giving a positive opinion in that line, because there's new data that is coming up every day and it's seemingly positive.
So the audio quality is not so great for me to understand at the moment, and I think it was Cynthia speaking. So perhaps Cynthia, if you could try again, because I tried, but it was not so easy for me to understand. Thank you.
So I was saying,
This is
considering
better.
that
Thank you.
data that has been brought forward in the recent years, regulators would actually consider giving a positive opinion from the one that was given in 2016.
And do you have some arguments why you think that perhaps they would give a different opinion? And also, just to clarify, at that time there was a divergent opinion, but in the end, overall, by majority, a positive opinion was provided, and also the medicine received a marketing authorization. So from that time already onwards, it received a marketing authorization and was made accessible for use in Europe. So already at that time it was positive, although some members had a divergent opinion. But if you could maybe give some arguments why you think that regulators today might take a different might all have a positive opinion, then that would be useful. Thank you. Any reflections on what the current regulatory thinking is about comparative efficacy testing for biosimilars?
A bit we have been discussing that last week, no? So it's a bit of a recap of what was being discussed last week.
I hope I got the question correctly. So the question is, what would be our opinion? Let's say in 2025, what would be our opinion with everything that is there? So I think she had brought out a good point that yes, we do have Remicade on the market, and probably what we'd have to do is, since our major concern was the studies in our ethnic group, what we'd have to do is that we would have to check what studies were provided and would then consider giving a positive opinion, and then would have to follow up with post-market surveillance. That's our response. Unless my colleagues want to add something.
Thank you, Prudence. I also see a hand raised by Birka, please.
Yes, thank you. I think it's a very interesting and very challenging discussion, what we will see here. When the colleagues asking for ethnic additional studies, and you have Remicade on the markets in several times, do you ever have seen that there are some influences due to ethnic considerations on the efficacy and safety of Remicade to substantiate or to justify your request for additional ethnic studies?
Yes, so as I said earlier, I think she brought out a good point that we have that product on the market, that's Remicade, which they were demonstrating biosimilarity. And if we have it on the market and we do not have any issues on the safety and efficacy, I think we, this is Mfaweli. If we did not see anything post-market surveillance for the original product, then I think our request then would not be justified in this case. That's why I stated that we would have then to give a positive opinion, because we're comparing it to a product that's registered and biosimilarity has already been demonstrated.
Thank you for the clarification.
Thank you, Prudence. Thank you also other students. Any other questions from anyone else in the group?
I have a question not on specifically on the biological issue, but on the comment regarding the voting system. And the background is a bit like that in Europe, obviously, we have many different countries sitting around the table in the regulatory submission discussions. And that might be also a point for discussion then for the future African Medicines Agency. Depends on how the system will be finally set up. But one needs to come to an agreement between different regions, different countries on a certain product. And I just wonder what else can one do if there are differences in opinions on how to interpret some scientific data. And if the voting is not an appropriate process, how any comments on what would be an alternative? Anyone from the team?
It's, we would then, I think, even though we thought that the voting system is a bit biased, I think we are also of an opinion that I know there can be different views and different opinions. Then I think there should be then an allowance that the final decision will be at country level. So each country will then have their final decision on the particular product, because I don't see any other way out where you have different opinions.
Regina, any comments from your side on that or another issue? Regina, you raised your hand.
Yes, thank you. And I'm very sorry, I'm struggling with my device. Thank you very much. Good afternoon, everyone. Adding on to the discussion, not about the voting system, but on the biologics and the biosimilar question, approvability, given the lack of data in the in the very population. I would just like to add to Birka's question, if you don't have any signals from the originator product as yet, when you decide on approvability of the biosimilar, I mean, is it entirely reassuring? The question, because my question is, isn't this also a question of the quality and of the post-marketing data that you get in your country, and the collection of post-marketing data, whether you see a signal or not? So it's a bit provocative, perhaps, the question, but I think only because one decision has been taken to approve the original biotherapeutic product, does it necessarily mean that for biosimilars there's never an issue? And I'll stop here. Over. Thank you very much.
Answer, because I think we need to close the session now.
I have a question. To the
Can we move that to another session? Because I think we need to close this session now for the sake of time.
Okay.
But if I would allow the final short, very 2 sentences response to Regina's comment, which was around how do you generate data, and would it be possible to generate sufficient amount of data to reassure about the safety efficacy and especially safety profile in a certain region?
Okay, so for our country, we do have a unit that is that specifically looks at post-marketing surveillance. So I think maybe at the time we were looking at this, we had not really gone a bit further to check with the unit how the data has been collected. But we are confident with the information that we get from the unit with regards to these other conditions or products that we get. So we would still look at the post-marketing surveillance data from our unit. And then normally we also refer to other reports in the SADC region if they are there. That's my contribution.
I think that there's a lot of food for thought and could raise and be a topic for a whole conference. But for the sake of time, we are not doing that conference now, but move over to the next presentation, which will be group number 10. And that will be a recorded presentation as agreed with the team before. And would like to ask whoever will present this recording to do this, and then have a discussion with whoever from the teams there. And Stephanie, we cannot hear anything.
Good afternoon, everyone. We are Group 10, and we are excited and eager to present to you our findings on KanGT, a trastuzumab biosimilar. My name is Nobulali, and I'll be joined by my clinical assessor colleagues, Nadine and Tato. To start off the presentation, we have to analyze what trastuzumab actually is. It is a monoclonal antibody that is used to treat certain types of cancers, mainly HER2 positive, which is human epidermal growth factor receptor 2 positive breast, as well as stomach cancers. It is found in intravenous and subcutaneous formulations. What exactly do I mean by monoclonal antibody? Monoclonal antibodies are a type of proteins that are produced in the lab that bind to specific targets on the surfaces of cells. These antibodies help the immune system identify and destroy cells or block the function of the targeted protein. How exactly does trastuzumab work? It works by binding specifically to extracellular domain of the HER2 receptors, and this binding blocks the receptor's ability to receive growth signals. By blocking HER2, trastuzumab inhibits downstream signaling pathways that promote cellular growth and survival. Trastuzumab also works by recruiting immune cells to attack the cancerous cells through a process called antibody-dependent cellular cytotoxicity. This enhances the immune system's ability to destroy the cancerous cells. Is this fantastic drug available on the South African market? Yes, it is, both in the public and the private sector under the brand names Herceptin in the subcutaneous formulation as well as the IV formulation. Trastuzumab is covered by most medical schemes, but coverage varies depending on the medical scheme. The financial implication significantly limits access to this drug. In 2017, trastuzumab was approved to be included in the essential medicines list, but is currently not on tender, thus limiting access as it is only approved for use for early stage HER2 positive breast cancer as a 6-month regimen. This illustration is to show cancer reoccurrence of conventional treatment, that being surgery, chemotherapy, as well as radiology or radiography, versus conventional treatment with trastuzumab in early HER2 positive breast cancer. In 2013, the EMA published a European public assessment report of trastuzumab with a clinical study of around 1,700 people had taken place. As shown in the graph, the blue is indicative of patients who had a reoccurrence of cancer while treated with trastuzumab as part of their therapy. And the orange is that of patients who had only received conventional treatment, that being chemotherapy, surgery, and radiography. The graph comparison on the left is of patients within the first year of treatments, and it showed that patients who were treated with trastuzumab, only 8% of those patients had cancer reoccurrence in comparison to the 13% of patients who did not receive trastuzumab as part of their therapy. The second graph comparison is over a 3-year period, where only 4.8% of the patients who had received trastuzumab as part of their therapy had cancer reoccurrence, while 11.8% of patients who did not receive trastuzumab as part of their therapy had the cancer reoccurrence. That is a 7% difference, which is a total of around 120 people who could have had, whose quality of life could have been improved by being simply put on trastuzumab as part of their therapy. The study also showed, after the 3-year mark, that 65% of patients who had received trastuzumab as part of their therapy were alive without their cancers worsening or reoccurring, whereas only 52% of the patients who did not receive trastuzumab were alive without their cancers worsening. This showed effectiveness in reduced risk of cancer reoccurrence, thus improving clinical outcomes for patients with HER2 positive cancers. Trastuzumab was also tested a study was also conducted of trastuzumab against patients with metastatic breast cancer, and it offers significant benefits over conventional treatments for patients with HER2 positive metastatic breast cancer. It has shown significant improvement in overall survival rates and progression-free survival compared to patients receiving chemotherapy alone, and aids in extending the time patients live without disease progression. Trastuzumab has also been shown to be effective in shrinking tumors in HER2 positive metastatic breast cancer patients, leading to better outcomes and inherently making surgical interventions more effective because of its targeted approach. I will briefly discuss the critical quality attributes of a biosimilar, which include structural similarities, which involves the primary amino acid sequence, which must be identical to that of the reference products, as well as the higher order structures, such as the secondary and tertiary structures. The secondary and tertiary structures refer to the folding and three-dimensional shape and higher order assembly of the protein, which is essential for functional activity. Any significant difference in structure could lead to differences in pharmacodynamics, efficacy, and safety. When it comes to biological activities, a biosimilar should prove its ability to elicit the desire of a biological response. Functional assays such as binding affinities, cell-based assays are used to confirm that the biosimilar retains the same mode of action as the reference product in a biological context. When it comes to purity and impurities, we assess a process-related impurities like host cell proteins and DNA, as well as product-related impurities like degradation of the products, using techniques such as the HPLC, as well as size exclusion chromatography. These impurities or degradation of products can affect safety, efficacy, and tolerability of the biosimilar. When it comes to pharmacokinetic and pharmacodynamic studies, the biosimilar should have a similar pharmacokinetic profile, as well as pharmacodynamic effects to the reference product to ensure comparable safety and efficacy. Stability studies are necessary to ensure that the biosimilar maintains its quality, safety, and efficacy over its shelf life. This includes testing under various storage conditions to evaluate the product's physical, chemical, and biological stability. Our product today that we'll be discussing is KANJITI, and I would like us to briefly speak on it and the tests that have been and how it performed on these tests that I've just elaborated on in comparison to the reference product. KANJITI is currently not registered in South Africa, but it is a proven biosimilar of Herceptin, which is registered in the UK. So how did KANJITI perform on these key tests? When it came to the structural similarity, it showed high similarity to Herceptin in terms of the amino acid sequence and higher order structures. Analytical studies confirmed that the structural attributes of KANJITI were comparable to Herceptin in terms of structure and size. When it came to the biological activity, KANJITI was found to be highly comparable to Herceptin, indicating similar binding affinities. KANJITI also showed very high purity levels with no significant impurities detected and remained stable over time. Stability took place under various conditions, and it was maintained, including refrigeration storage, as well as in use temperatures. KANJITI maintained its quality and bioactivity throughout these temperatures. The rationale of approving KANJITI based on these results. KANJITI was approved based on its comprehensive testing and demonstrated comparability with Herceptin across the critical attributes, which were the safety and efficacy, where the trials showed that KANJITI had no clinical differences in terms of safety, purity, or potency compared to Herceptin. In terms of its pharmacokinetic profile, it showed high similarity to that of Herceptin, meaning that the drug behaves the same way in the body as Herceptin. When it came to the immunogenicity, which is the potential to cause an autoimmune response, it was comparable to Herceptin. And structurally, these involved extensive analytical testing that were done to demonstrate that Kanjinti is highly similar to Herceptin structurally and functionally. And based on these results, collectively, these results supported Kanjinti to be approved, as it was safe, effective, and of quality as Herceptin. I will now hand over to my colleague, Nadine, to take us through the clinical pharmacological data.
Thank you, Nabulali. I will be providing our opinion on the comparative pharmacokinetic data. The table that you see reflected is an extract from the EPA report that tabulates studies that were carried out prior to the marketing authorization application. 2 studies have been reported. The clinical pharmacokinetics program comprises a pharmacokinetic similarity study conducted in healthy subjects using a single 6 milligram per kilo IV dose. The clinical similarity study included the collection of pharmacokinetic data in subjects with HER2 positive and early breast cancer. A pivotal phase 1 study demonstrated that Kanjinti is bioequivalent to both EU and US source Herceptin. The randomized single-blind, single-dose, 3-arm parallel group study involved healthy male subjects and evaluated pharmacokinetic parameters such as the area under the concentration time curve from 0 to infinity and maximum serum concentration. The results showed that the 90% confidence intervals for the geometric mean ratios of these parameters were within the bioequivalence criteria of 0.8 to 1.25, confirming similar pharmacokinetic profiles between Kanjinti and the reference product. In the clinical similarity study conducted in women with HER2 positive early breast cancer, the dose and frequency of dosing were chosen in consideration of the current trastuzumab prescribing information. When analyzed by treatment received in the neoadjuvant phase for subjects in the pharmacokinetic analysis, population results showed that pre-infusion trough concentration was similar between the test and reference product in all visits during the neoadjuvant phase. For the subset of subjects where additional post-infusion serum samples collected, results showed that post-infusion levels of the test and trastuzumab were comparable. In addition, a population pharmacokinetic analysis using published population pharmacokinetic models and the observed data from the phase 1 pivotal study was performed to assess the consistency of the pharmacokinetics of the test with the pharmacokinetics of trastuzumab reported in the literature. The estimates for all fixed and random effects parameters were highly consistent with reported pharmacokinetic parameters as reported in literature. A single electrochemiluminescence assay in microtiter plates was designed to quantify the test and reference product in human serum. A pharmacokinetic method qualification study was performed using independent standard curves of both the test and reference source Herceptin. Full validation results met the criteria as per the guidelines for bioanalytical method validation. For both studies, samples were evaluated for binding anti-drug antibodies using a 2-tiered immunoassay that consisted of a screening assay and a confirmatory assay. For both studies, samples were evaluated. A validated ECLPG immunoassay was used to detect antibodies capable of binding the test and reference product. Hence, the data is collected and processed has been done via validated methods aligning with prescribed literature and similarities of results between the test and reference product. The pharmacokinetic studies are considered sufficient. Kanjinti was tested in a specific indication as described in the EPA for early breast cancer. The selection was done of the sensitive and homogeneous population. The most sensitive, meaning that they're earlier in the disease, generally more in a healthy state, less disease burden, and have less prior lines of medication. All indications approved for the reference product Herceptin. and scientific evidence indicate that the mechanism of action of trastuzumab is similar in different target conditions in both early and metastatic breast cancer, as well as HER2-positive gastric cancer. Hence, resulted in an extrapolation to the non-studied oncology indication, and these were considered acceptable. The extrapolation of indications were for metastatic breast cancer as well as metastatic gastric cancer. The endpoints chosen for the studies and its validity is the pathological complete response rate in breast and axillary nodes, regardless of ductal carcinoma in situ as primary endpoint. This was accepted by CHMP owing to the fact that the secondary endpoint included the same endpoint, but in the absence of ductal carcinoma in situ. The co-primary efficacy endpoints included risk ratio and risk difference of the incidence of PCR in breast tissue and axillary lymph nodes, while the secondary efficacy endpoints included risk difference and risk ratio of PCR in breast tissue, as well as risk difference and risk ratio of PCR in breast tissue and axillary lymph nodes in the absence of PCIS. The use of pathological complete response as a primary endpoint is considered valid and appropriate for several reasons, some of which it has a vast regulatory acceptance. Regulatory agencies, including the European Medicines Agency, recognize PCR as a meaningful endpoint in neoadjuvant breast cancer studies, particularly for assessing the efficacy of treatments targeting HER2-positive tumors. The predictive value achieving PCR has been associated with improved long-term outcomes, such as disease-free survival and overall survival, especially in aggressive cancer subtypes. And the feasibility. PCR allows for a more rapid assessment of treatment efficacy compared to waiting for long-term survival data, facilitating timely regulatory decisions. Our recommendation would be to approve the drug based on the pharmacokinetic studies. Both studies demonstrated similarity. Pharmacokinetic studies, and in addition to the efficacy, safety, and immunogenicity study, is sufficient for approval of ClinReg2024. As aligned with the EBA, phase 4 studies can be requested, however, still maintaining that the drug should be approved and be made available to the market. And now hand you over to my colleague, Putato.
On the differences in administration of medical practice in low resource settings, the key considerations that must be considered is the availability of skilled and trained healthcare professionals to administer IV infusions or subcutaneous injections, resource and infrastructure constraints. So some facilities may not have adequate access to storage, handling and administration equipment for IV infusions. Patient access. So some patients may have difficulties accessing healthcare facilities or may require more convenient treatment options. And then on cost and affordability, the cost of treatment, including administration costs, may be a significant barrier for patients in low resource settings. Highlighting the differences between ClinReg2024, the biosimilar, and Herceptin, the originator. With ClinReg2024, it is approved and administered for the IV infusion group only. The IV infusion administration may be prone to errors. There might also be increased healthcare costs as there's an increased need for trained personnel and advanced equipment. There's less variety of settings for application. And then with Herceptin, it's approved for subcutaneous injection as well as IV injection. The subcutaneous route might be more suitable for low resource settings as it requires less equipment and training. And with regards to access, the subcutaneous injection route will increase access to the ease of administration. So there were two opposing views on the use of ClinReg2024 from CHMP, which is the committee in EMA. The majority view was that there was a strong similarity in quality, where ClinReg2024 was considered to be similar to the originator, as it demonstrated similar quality characteristics to Herceptin. So the quality data demonstrated that the acceptance criteria was met in a narrow range. It also indicated a robust and controlled manufacturing process. The batch analysis that was presented also supported this conclusion. The proposed control strategy for Kanjiti was also considered to be appropriate. The other reason was that there was comparable efficacy and safety, as extrapolation to the non-studied oncology indications was considered acceptable, as scientific evidence had demonstrated that the mechanism of action of trastuzumab is similar in different target conditions like in early and metastatic breast cancer and gastric cancer. The other reason was that there was no clinically meaningful differences between Kanjiti and Herceptin, and although small numerical differences were noted, such differences were considered to be clinically insignificant because the overall observed safety profile of Kanjiti and trastuzumab were consistent with the historical safety profile of trastuzumab. So members that held the majority view believed that the totality of evidence where similarity supported by quality, non-clinical, pharmacokinetic, as well as safety and immunogenicity data demonstrated similarity in terms of efficacy. Then a few CHMP members expressed a divergent view, arguing that there was insufficient clinical efficacy data, where one study failed to demonstrate the equivalence in PCR rates according to pre-specified equivalence margins, and explorations of the relationship between ADCC activity of clinical trials batches which have been hypothesized to impact efficacy and PCR rates were considered to be inconclusive. Therefore, they felt that biosimilarity had not been demonstrated. However, the conclusion was that the CHMP adopted a positive opinion due to the overall benefit-risk ratio, which was positive. So the possible reasons for the variable outcomes of the clinical study might be due to many factors. One might be product specific. So differences in manufacturing processes, formulation, or analytical testing differences, or differences in endpoints, whether it's primary or secondary endpoints used between the biosimilars and the reference products, differences in the study design and the methods that were used, differences in the models, as well as the ways of interpreting data, and also differences in the patient that we administered the test medicine, which can impact treatment outcomes. So we feel that the variable outcome of the clinical study might be related to the biosimilar rather than the originator, due to the fact that the originator already has an established safety and efficacy profile with extensive clinical experience and a well-characterized pharmacological profile. And due to the fact that we also know that there's a lot of challenges with biosimilar development, where small differences in the manufacturing process, the formulation, or the testing will impact its performance. So here we had to summarize an article that we read. The article highlighted the analytical characterization of the biosimilar and the reference product is an important step in the development process. So in the study, Herceptin was characterized using multiple batches where inconsistencies were observed in the test results. They highlight the differences in ADCC, where drifts in ADCC related quality attributes were observed amongst the batches. So Herceptin illustrated two marked drifts in its ADCC-related quality attributes, showing a downward drift in alpha-cosylation levels and an upward drift in high mannose level. So the drifts affected the development of the trastuzumab biosimilar, as the manufacturer had to exclude the affected batches of Herceptin from their analysis to ascertain the target quality ranges for their proposed biosimilar. The article outlines the importance of monitoring the quality attributes of reference products throughout the biosimilar development process to ensure that the biosimilar continues to meet the required standards for quality, safety, and efficacy, and just ensuring that there's extensive analytical characterization that is done prior. So our approach on assessing divergent data that might come up on originator and biosimilar is first to identify the cause for the inconsistent data, and from there assess the impact on biosimilarity by evaluating whether the changes in Herceptin's quality attributes affect the biosimilar's ability to demonstrate biosimilarity, to assess whether clinical trials and the studies that were performed are comprehensive and robust enough despite the changes in the quality attributes, to also request additional data or studies to clarify the impact of the changes in the quality attributes of the biosimilars quality, safety, and efficacy. So our recommendation is to grant approval with conditions for the biosimilars applicant to conduct additional studies and monitoring. So on adopting reliance for quantity assessment, applying the reliance pathway might work provided that the sufficient and reliable data that demonstrate quality, safety, and efficacy, and there are adequate risk management plans to address the potential risks or uncertainty. So the benefits to applying reliance in this case would be that there's increased access to life-saving treatment. So applying reliance will lead to an expedited review process, leading to faster approvals and access to treatment. There will be a reduced need for clinical trial studies, which use a lot of resources, and the resulting effect would be that there will be a cost benefit, which will translate to patients also, so they will be able to access the drug at a cheaper rate. And then the limiting factors is that some regulatory agencies do not publish their public assessment reports, so there's lack of transparency, and there is sometimes difficulties in obtaining unredacted reports from recognized regulatory authorities that register the product. There's also region-specific requirements that every regulatory authority must still abide to, even if they are aligning to another RRA, which might limit the use of the reliance model. So our key learnings from this case and just from biosimilars discussion, we appreciated learning about the complex science of biosimilar development. We recognize the need for comprehensive regulatory frameworks to assess biosimilars, the need for capacity building to address the critical knowledge and skills gap that exists. So this program aims to address that, the benefits and challenges of adopting reliance pathways and harmonization initiatives to facilitate biosimilar assessments, enhancing patient access to affordable treatment through biosimilars. So as Group 10, we hope to use these learnings to spark meaningful discussions on biosimilar and also just explore ways in which we can all contribute in the biosimilar space, especially in Africa, where they would make a significant impact on the lives of people. Thank you.
I would like to thank all the presenters in this pre-recorded presentation, which was pretty comprehensive. Unfortunately, it took all the half an hour, so that we have no good time now to go into a discussion. What I would like to do is to ask Lise to give a brief assessment of what has been said, so that nothing keeps hanging in the air, but not go into a long and lengthy discussion for time reasons.
Great, thanks. And also from my side, thanks for the very comprehensive, detailed presentation, and also for sharing your personal learnings and reflections at the end. I might ask you one summarizing question, or maybe two if there's a little bit of time, but let's see. First, could you explain to me and to the group, you look both into the dossier of the originator and into the dossier of one of the biosimilars for trastuzumab, what are main differences between a clinical efficacy trial that you would see for an originator and that of a biosimilar in terms of purpose of the clinical trial and also in terms of study design choices? Could you share some reflections on that, please?
Hi, Lise.
Yes, thank you, Nadine.
So from our side, I know when we looked at it initially, so we looked at Herceptin that's been registered for Roche. We did find differences in the clinical endpoints of the clinical studies that were shown. So from the initial application for the originator, we found that the clinical endpoints were completely different to what we what we saw in the Kenjinti one, whereas they had like time to progression as a primary endpoint and the safety profile of Herceptin, as well as like overall response rates. On our side, we had the PCR is a clinical endpoint. But what I did see in there as well was that there was also an extension of the initial application of the originator for a new route of administration. And it was initially approved for the solution for infusion, but then applied for like subcutaneous injection. And actually, in that one, the co-primary endpoints were one was pre-surgery, and the other one did include the pathological complete response rate. So definitely more clinical studies done on the originator long before submission, whereas on the biosimilar, we only had the 2 clinical studies.
Thanks, Nadine. And indeed, you rightly mentioned that there are differences in endpoints that you observed between the originator trial and the biosimilar trial. Is this something that you expected? And could you maybe link it to the goal of the trial? Maybe both trials wanted to demonstrate something different, and that's why there was also a choice of different endpoints. Any thoughts on that aspect?
So for the originator trial, I think there are a lot of more safety data and efficacy data needed to be proven. And that was the reason for the selections in their endpoint. Whereas, for example, like the duration of response, something even like quality of life was used as a secondary endpoint. Whereas in the biosimilar clinical study, I think because of the time frame in getting this to market, a more feasible endpoint using PCR, because your data is attainable in much shorter span, was selected.
Yes, thank you. And perhaps to add to your answer, indeed, for a new active substance, we need to, for the first time, demonstrate efficacy, safety. So, for example, for oncology medicine, you will then oftentimes see overall survival or progression-free survival as endpoints, where for a biosimilar candidate, we don't want to, again, demonstrate its standalone safety and efficacy. We want to do it in a comparative manner to the originator. We want to be able to demonstrate that it is as safe and as effective as the reference product. And for this, we need endpoints that help us to demonstrate that goal. And for this, we then select endpoints that are generally more sensitive to potentially capture minute differences between biosimilar and reference products. So that's why for Kanjinti, you then saw, for example, that they went for a pathological complete response, because it's more easy to measure, also for small differences, and also indeed more easy to measure shorter in time. So we always need to reflect back, what is the goal of this trial? And for a biosimilar, it's not this demonstration de novo of safety efficacy. It's about demonstrating biosimilarity, of which the clinical comparative efficacy study is one point of the overall totality of evidence that regulators look at. Maybe one more question, but I leave it up to Bernd to decide if there's time for that or not. Thank you.
I have just made in-house for myself decision that we go 15 minutes overtime at the end of this day, so until 5:15. So we would have another 2 minutes for discussion, but then we need to close this session.
Okay. Thank you. So maybe a second question from me for the group is, so you discussed this case of trastuzumab, both information about originator and also a biosimilar. And you saw indeed for the biosimilar that there was also divergent opinion, because it had a convincing biosimilarity package based on the analytical functional comparison, also on the level of the comparative pharmacokinetic study. But there was some doubts about the comparative efficacy study. You still also mentioned in your presentation that you would also have a positive recommendation. I was now wondering if it would be an opposite situation. So you would still have very good comparative functional and analytical data. You would have also a positive comparative efficacy study, but you would have a failed comparative pharmacokinetic study. What would be your opinion in such situation, and why might it be different compared to a failed comparative efficacy study for a biosimilar candidate? Any thoughts on that? Very welcome. Thank you.
Sorry, I'm not sure if I'm audible, if I should write in the chat, but I think just based on the fact that we deduced that there was no clinically meaningful differences between the biosimilar as well as the originator, as well as all the other studies that were conducted from the comparability exercises. So that made up a big portion of why we also agreed with the positive recommendation. But our concern was mitigated with the fact that we're going to give approval, but also just request additional data or monitoring to be conducted.
Thank you. And here my question was also about a hypothetical situation that in the dossier that you would have had done the assessment on would have contained a failed comparative pharmacokinetic study between originator and biosimilar, and how that would influence your decision making. If it would make you lean more towards negative, or if you would say same as here with the failed comparative efficacy study that you saw, okay, given the fact that we have overall a strong totality of evidence for biosimilarity, we still lean towards positive opinion.
So Liz, if I could add on to what Kotatsu's just said, I think in terms of failing to meet the bioequivalence criteria from a PK study, we need to investigate what was the reason for the failure, and definitely it would be a delay in approval, and it would sway the decision. It may not be the be all and end all, but it will definitely delay the regulatory approval, and we can then look into it, or consult with the applicant in terms of what was the root cause analysis for it. Was there any flaws within the study design that could probably have caused any the PK failing? Because if it's been proved similar in every other facet, then it should be meeting comparable standards as the originator.
Thank you, Nadine, for that clarification. And indeed, a comparative PK study would be a basic requirement. It would really be a prerequisite. And in terms of weight of evidence, it has more weight to it in the overall totality of evidence for biosimilarity assessment, because it's a more sensitive tool to potentially detect or pick up on potential differences. So indeed, it would be very crucial to have a successful comparative PK study, where for the comparative efficacy study, we see that the requirements are decreasing as it's a less sensitive tool. So giving less weight to it in the overall assessment. Thank you. Bernd, over to you.
Thank you. I think that was a very important discussion and additional learning exercise, and it was useful, I think, also that to have that extend that discussion. And as mentioned earlier, and also in the chat, we will finish at 5:15 South African time, so we make it a bit longer. But we also had some technical issues with one of the presentations, so that will allow for this. The next presentation will be by group number 11, which is Tirani, Ziyeng, and Fuyelwa, with another example, which is pegfilgrastim. We discussed that already a bit, but was already mentioned last week. And now we go a bit more into the discussion on that topic from now from that group. Looking forward to the presentation.
Okay, thank you. Good afternoon. My name is Tirani, and in Group 11, I'm with Dora Diale and Viola Ndoyeke. So we are looking at Palmec, which is the biosimilar of Neulasta. So I'm going to hand over to Dora to cover part 1.
Yes, good afternoon, colleagues. I will be looking at part 1, the clinical necessity of the medicine. And we have been asked a few questions, so we'll just go through them. The first question is that, is it responsible to approve a biological product without underlying trial on efficacy and safety in patients? So our response is that we sort of looked at both the requirements for an NCE and the biosimilar. So with an NCE, all the new the NCE's entities to be utilized by the general public needs to have demonstrated efficacy and safety in both animals and humans through the conduct of clinical trials. So the NCE cannot be approved without the underlying clinical trials, and this is really to reduce an element of uncertainty. So ordinarily, NCEs would be done through all phases of clinical trials, but this will depend then on the type of disease under investigation. So for biosimilars, because the clinical trial in biosimilars has a different goal than as in NCE, the goal for biosimilar development is to demonstrate similarity and not the clinical benefit, as this would have been proven or demonstrated from the parent molecule. However, the same high standards of quality and safety and efficacy needs to be applied throughout. And a detailed comparative exercise is required with the goal of demonstrating that the biological medicine has a similar quality attributes before and after manufacturing process changes as that of the reference product. Again, the authorization of biosimilars requires a different set of data compared to that of the reference product. For biosimilars, there is no need to de novo establish clinical efficacy and safety, which is well known for reference product. In general, the requirement is that one comparative, this is for simple biologics, that only one comparative PK/PD study in healthy volunteers would be sufficient using a validated biomarker. And for complex molecules, then additional study would be required of the safety and efficacy to test one of the indication in very sensitive population. And so in response is that it is acceptable to approve a biosimilar product without underlying efficacy and safety trial in patients. Next slide, please. Yes. So the second question for us is to look at the availability and reimbursement of filgrastim in South Africa for the supportive treatment of cancer patients. Yeah, we sort of looked at the requirements in the EML. Filgrastim is registered in South Africa by Amgen as a Neulasta originator and with 6 generics. However, it would seem that only 3 generics are marketed because there's an assumption because they do not have an SEP price, single exit price. So from the EML, we found that only 3 generics were available in the market for use by patients. And in South Africa, filgrastim is not on the EML, but filgrastim is. So in the public sector, the availability of filgrastim could be obtained through a named patient motivation process, possibly using a special tertiary grant approved by respective pharmacy and therapeutics committee. In the private sector, usually the funders would conduct the health technology assessment. They will have their own list of medicines, and they could decide based on the outcome of that HTA, they could opt not to reimburse or to reimburse in full, or co-payment would apply. And the benefit of having generics is that it increases access for patients in the healthcare system and saves the cost for the healthcare system as well. So the third question was to look at the net benefit of filgrastim over filgrastim in terms of patient outcomes. And we sort of looked at the pharmaceutics of the product and social part of it, that filgrastim is given subcut, single subcut after each chemotherapy cycle, whereas filgrastim is required up until the expected neutrophil count has recovered to the normal range. So that means that it will require a multiple dose as compared to filgrastim. And the downside of filgrastim is that, especially for IVI, is that it increases the number of hospital visits, it increases patients and hospital burden, thereby affecting quality of life. And there is potential loss of income for missing work days. And it can become costly for a patient, who is somewhere in deep rural areas, who would require transport and meals for hospital costs. And filgrastim has a long half-life compared to filgrastim. Adherence may be an issue for filgrastim. And there are two articles that we sort of looking at the difference between the two, and it seemed that there is no major difference, but the article is in favor of using pegfilgrastim. Thank you. Next slide, please.
Okay, so we're getting to the second part of our presentation, which looks at the physical and chemical quality data. And the question was, what do you think are the 2 or 4 most critical quality attributes that need to be assessed for a biological medicine like pegfilgrastim? So we looked at 4 of those, which is physiochemical similarity data, and the biofunctional similarities, the stability testing to demonstrate the degradation behavior of the product, as well as the degradation profiles. And for the physicochemical similarity data, we looked at the comparison of these parameters, which is your primary structure, your intact mass and petulation, higher order structures, purity and impurities, looking at the product-related variants, and also some other impurities, same as also the general properties. And under biofunctional similarity data, looking at the parameters to compare this, which is potency as well as receptor binding. And for stability, looking at the accelerated and stress stability and long-term stability testing, and looking at those parameters and doing the similarity comparison, as well as the degradation profile, which looking at the stress stability. And the next question was, how did PELMEG perform in these tests? And what are the critical quality attributes that were used? So the biosimilarity between the PELMEG and the Neulasta on these critical quality attributes had the following outcome. They're both delivered in identical formulation and same presentation, with the exception of the pH. So PELMEG pH is slightly higher than that of the Neulasta. However, it has no impact on the structure and the biological function of the PELMEG. And also looking at the analytical tests that were conducted, it proves that PELMEG is highly similar to that to Neulasta. And looking at the purity parameters that were looked into, it's slightly different, but with no impact on the product quality. And with a primary structure of the two, they have identical sites of petulation. Molecular weight, they both have similar peg moieties. And looking at the higher order structure, which is product-related variants, and the impurity as well as aggregation profiles, they are similar. Question is, would you recommend approval based on these tests? And our response is that yes, because then the data of the physicochemical, the biofunctional, as well as the stability test confirm that from a quality point of view, the PELMEG could be considered as biosimilar of the Neulasta. I'll hand over to Derani to look at the third part of the question. Thank you.
Thanks, Vuyelwa. So I'll be taking us through the clinical pharmacological data. And the first question was to share our opinion on the comparative pharmacokinetics data. So looking at PELMEG, pegfilgrastim, our opinion was similar to that of EMA, that the development program for PELMEG was adequate. It was performed in line with guidance for similar biological products, and they had incorporated scientific advice that they received. The program included studies in healthy subjects. The population was considered to be the most sensitive for PKD comparison versus cancer patients in chemotherapy, simply because healthy subjects do not have comorbidities. They are no concomitant medications, and they do not have immunosuppression. So the sensitivity and homogeneity of the study population was therefore suitable to reduce variability and co-founding factors. Also, how the studies were designed in the program, looking at the long half-life of pegfilgrastim, they had a 6-week washout period, which was essential to ensure that critical parameters are re-established, are re-baseline before the crossover could occur. The crossover design also addressed the expected high inter and intra-individual variability for PEC. Next slide, please. So the indication that was tested for PELMEC according to the IPA report. So the reference product only has one indication, which is the reduction in the duration of neutropenia and the incidence of febrile neutropenia in adult patients treated with cytotoxic chemotherapy for malignancy. So that was the indication that was tested for PELMEC as well. There were no dedicated comparative efficacy trials considered necessary because pegfilgrastim is a biological with a less complex structure. Therefore, the pivotal evidence could be derived from the similarity of the physicochemical functional properties as well as the PK and PD comparisons. In addition, the CD34 and AA absolute neutrophil count AUEC were tested in healthy volunteers as secondary endpoints, which therefore provided supportive data for efficacy similarity with the reference product. Next slide, please. And then looking at the endpoints that were used in human studies and confirming their validity for drug availability. So there were 2 studies that were conducted for PELMEC. The first study which was a pivotal PKD study. The primary endpoint was the AUC area under the curve from time 0 to last measurable concentration, the maximum concentration, as well as the area under the effect curve of the absolute neutrophil count. The secondary endpoints were CD34 plus cell counts, area under the curve from time 0 to infinity, the maximum, which then measures the maximum effect level, and the time to maximum effect level. The second study was a supportive study whose primary endpoints were area under the effect curve of the absolute neutrophil count, and the secondary endpoint was the time to maximum effect level and CD34 cell count. So in terms of validity, we think the endpoints could be considered valid because when you have to confirm efficacy for biosimilarities, you need to look at clinical equivalency on the basis of pre-specified margins, along with comparable safety and immunogenicity, which were proven to be similar for PELMEC versus the reference product. So the similarities, which were within the prerequisite equivalence margins of 90 to 111% with 95% confidence interval, which, so these parameters are from the bioequivalence guidelines, were on the drug concentration, the half-life of the drug. And in addition, the pharmacodynamic data and immunogenicity properties evaluated as surrogate markers of clinical efficacy and support of biosimilarity of both products. So therefore, the primary endpoints in both studies were met, providing sufficient evidence to the similarity between PELMEC and this reference product. So in terms of our recommendation on the overall package, the similarity has been demonstrated between PELMEC and the reference product, and therefore approval will be granted with the following recommendation for the applicant to consider. To include risk minimization strategies and ensure updating of the SNPC and labeling as the information becomes available, and continuous monitoring of adverse events in clinical practice and active reporting of those adverse events to the regulatory authorities. I'll hand over to Dora.
Thank you, Tirani. So I will be presenting now part 4, which is clinical critical appraisal. Next slide, please. And we needed to look at the number of patients involved in studies included in the submission package. We found out that for a pivotal study, 172 patients, that is healthy volunteers, they were enrolled into the study where they've assessed the PK/PD as core primary endpoints. And for the supportive study, then they enrolled 96 patients and assessed the PD and immunogenicity as core primary endpoints. Next slide, please. So is it essential to require studies in patients before approving every biological product, the position in SA? So we sort of looked at the difference between the EU and South Africa, and the EU really does not recommend conducting all studies in patients before approving every biosimilar product, and clinical trials for biosimilars do not need to include the pivotal studies conducted for the reference product to prove safety and efficacy in humans. For single chain, as highlighted before, is that one comparative PK/PD studies in healthy volunteers would be sufficient. And for complex molecules, one additional comparative safety and efficacy study in sensitive population will be required. And also confirmatory clinical trials with biosimilar are usually not needed for every indication that has been approved for the reference product. So the extrapolation in this case would be possible, especially where the target is the same. So we sort of looked at South African requirements, and the requirements are very, very similar. Although South Africa does not prescribe the number of studies that needs to be conducted, but the requirements are quite similar. Next slide, please. Okay. Can you go to the next slide? Okay. The next one is that we needed to look at the UK policy. Cases where a patient shall
quickly intervene. Could you gradually come to a conclusion so that we have some more time also for discussion?
All right, perfect, thank you. So we look at the UK policy, and UK policy states that the recommendation from the UK is that a biosimilar development needs to be on a case-by-case basis. There are principles that they have defined that needs to be included when making that decision. The first one is justification of a comparative, comparable efficacy. And the second one is justification for comparable safety and immunogenicity. And the third one, they highlight that the need for an efficacy trial will still be required, especially where there's lack of understanding of the biological functions of the reference product to its clinical effects. So there are cases whereby the studies would still be required. And next slide, please. I'm just trying to move very quickly. The next slide is to look at the value of patient trials in biosimilar approval. So that provides a framework to enable the marketing authorization and confidence that the product utilized by the general public has demonstrated biosimilarity, and also assurance that there is no clinically meaningful differences in terms of quality, safety, and effectiveness. When you have a good comparative pharmacokinetic studies in healthy volunteers, and that could help in extrapolating the data on safety and efficacy. And also it saves costs for further clinical trials development, and it further fosters competition and innovation, whereby it increases the access of medicines. And the other question is the number of efficacy studies in patients for Zaziro is a filgrastim. There are 2 studies here that were done, and the first one was in PK/PD, and the second one, they've done a phase 3 study where they looked at patients, breast cancer, high-risk patients, local advanced metastatic cancer. Next slide. So here we're comparing the striking differences between Zazio and Pelmec. So here, the striking differences is that Pelmec only conducted one study in healthy volunteers, whereas Zazio also is one study in healthy volunteers, including the phase 3 studies as well. So there are other slight differences, which for the sake of time, I may not be able to mention, but there are like in vitro and vitro studies that they have also highlighted in the report. Okay. And then opinion about applying reliance in assessing the product in South Africa, yes. SAPRA endorsed reliance, and there is a guideline for that as well. So we sort of looked at the benefits and the downside of reliance. The benefits of reliance is that it builds capacity for less mature regulatory authorities, and it reduces the burden as applicants compile a single dossier for submission to multiple countries. There's an opportunity for harmonization and convergence of regulatory authority processes. It decreases the cost of review as well, and it brings on efficiencies whilst improving the regulatory systems. And it allows for increase of number of biologicals to come into the market as well. It builds trust among different authorities, and access is a positive in this case, and also decreases the review timelines. And the disadvantage is that we've seen instances whereby if one country does not agree with a certain recommendation, there may be additional queries that comes as well, which delays the review process. There are also country-specific requirements that needs to be complied with. And also, sometimes there's no consideration for country original disease burden and no central repository for reliance information. So we have seen quite a few reliance products that goes through the Zazibona procedure. Thank you.
So lessons learned out of this is that for assessment of the biological products, there is no one blanket fits all approach in assessing them. Although some guidance is provided by the regulators, but each assessment would differ based on the size and the type of the molecule, the mechanism of action, the patient population, PK profile, the type of disease being investigated. To ensure that there are no challenges with the review and approval of the biosimilar, it is important for applicants to provide required data and consult with the regulators earlier on in the study to ensure that the endpoint in question are applicable to the design, the population, as well as the study being investigated. And thank you. That's the end of our presentation.
Many thanks also to this team for that excellent presentation, which addressed all the issues that have been raised in the question. And I would like then to hand it over again to Lise, 5 minutes for the discussion.
Thank you to Group 11 for the nice and very exhaustive presentation. I liked one of your key takeaways, and that's that there is no one size fits all for biosimilars, which is very true. There's not one type of biosimilar. Biosimilars can be developed for a wide range of biological medicines, for the more simple products, such as perhaps filgrastim, to the more complex monoclonal antibodies. So indeed, it always requires a case-by-case assessment. You mentioned in one of the last slides that for Zarzio, which is a biosimilar for filgrastim, you saw in the clinical data package both comparative PK/PD and a comparative study in patients, a comparative efficacy trial. While for Pelmech, you didn't see this. For Pelmech, for pegfilgrastim biosimilar, you saw only the comparative PK/PD study and no comparative efficacy trial. Could you perhaps reflect why you saw different levels of clinical evidence for these two biosimilar candidates? And perhaps you can also position it a little bit in time of when these products were evaluated. Thank you.
Can you please repeat the first part of the question?
So for Zarzio, which is a biosimilar of filgrastim, you saw a more extensive clinical data package consisting of both a comparative PK/PD study and comparative efficacy testing trial, where compared to Pelmech, you only had a comparative PK/PD study. Can you maybe explain why one dossier contained a comparative efficacy study and the other didn't? Has it something to do with the time at the time point that these medicines were assessed? And also today, what would you expect for a filgrastim or pegfilgrastim biosimilar? Would you or would you rather not expect a comparative efficacy study in the clinical data package?
Okay, can go to it. So yes, I think indeed it does have to do with timing, because if you look at Zarzio, it's a very old molecule, and at the time, I think maybe perhaps the regulators did not have much knowledge on the biosimilars, right? And it's one of the very first few biosimilars that was reviewed in EU. And So the current stance is that for not if for simple biological product, one comparative PKPD study would be sufficient. So going forward, in terms of the review, we do not expect to see the efficacy and safety trial being conducted for these products.
Great, thanks for the concise but interesting answer indeed. So the first product Zarzio was approved in 2009. So this was very early in the days of having a biosimilar framework and having the first biosimilar assessments, whereas Belmech was in 2018. So 9 years later, with already more experience. In the meantime, also EMA published to take the European perspective, a specific guideline on also biosimilar assessment specifically for these GCSF type of products that gave more detailed and concrete guidance on the fact that if there is a comparative PKPD study conducted, that a comparative efficacy study might be waived. But we see indeed for original biosimilar developments that sometimes a more comprehensive approach was chosen because there was also more uncertainty on the regulatory guidelines and also on what regulators would expect. And we see that regulators in response developed more guidelines, more specific guidelines on a product specific level, and that overall more experience has been gained so that also developers now more apply this more concretely to their developments to tailor their programs. Also in view of time, no particular additional questions from me for Group 11. Thank you.
Thank you very much. And that brings us again in time with a delay time frame. And the last team is now a similar topic, and that is Priscilla, Tonray, and Tracy. If there are any duplications in your slides versus the previous group, then please take the opportunity to go quickly through that, so you will not be penalized for being quick. But any additional thoughts, ideas that you have, are highly welcome. And with this, I would like to call the last group, which is group number 12. And we can see yours. Yeah, now we have it also in presentation mode. Please go ahead. And we cannot hear you. We see the slides, but we don't hear anything.
Hello, can you hear me now?
Yes, we can hear you, but
we can hear you.
Sorry,
It's very soft. And could you switch on your video if that's possible?
Hello, can you hear me?
If not, then just go ahead.
Okay, so I can.
Now you are muted, so we don't hear you.
Biosimilar for the reference product, which is filgrastim. So, pegfilgrastim with the trade name Pelmeg is indicated for the reduction of neutropenia and the incidence of febrile neutropenia in patients receiving cytotoxic chemotherapy for malignancies, except for chronic myeloid leukemia and myelodysplastic syndromes. So This medicine mode of action works through binding to the granulocyte colony stimulating factor receptors on the surface of progenitor cells in the bone marrow. This binding activates intracellular signaling pathways that promote the proliferation, difference, and survival of neutrophil precursor cells, leading to an increase in the production and the release of mature neutrophils into the bloodstream. So this slide is showing a comparative factor of pegmil and Neulasta. I'll go to the next slide. So our first question stated that is it responsible to approve a biological medicine without underlying trials on safety and efficacy in patients? So it is yes for biological similars based on the satisfactory pharmacokinetics and pharmacodynamic studies in youth participants. For example, in pegmil, the studies were conducted using absolute neutrophil count in surrogate endpoints for pelmage. So for innovator biological products, they have to go through clinical trials to confirm that they are safe, effective, and of good quality. Now, second question stated that is pegfilgrastim available and reimbursed in your country for the supportive treatment of cancer? So pegmil is available in Zimbabwe as a prescription preparation, and it was registered in 2019. It is available for free for senior citizens in public hospitals, but for anyone who is below 65, they can access it using their medical aids or cash. So our second question stated that what is the net benefit of pegfilgrastim over filgrastim in terms of patient outcomes? So pegfilgrastim has got similar effectiveness in reducing neutropenia duration and prevention of febrile neutropenia, and it also has a prolonged action, which may offer slightly enhanced outcome. Multiple clinical trials have shown that pegfilgrastim is at least as effective or superior in preventing febrile neutropenia compared to filgrastim. And also pegfilgrastim promotes convenience and adherence to patients because it requires a single injection per chemotherapy cycle because of its prolonged half-life. Additionally, pegfilgrastim reduces rates of neutropenia related hospitalization and decreases the need for antibiotics compared to filgrastim. Also, although pegfilgrastim has a higher cost purchasing price, fewer injections, lower hospitalization rates, and decreased antibiotic uses can lead to a cost saving in overall healthcare utilization. Moving on to the fourth question, we stated that critical quality attribute that needs to be assessed for a biological medicines like pegfilgrastim and filgrastim. So for the critical attributes that need to be assessed, we discovered that the primary structure and the post-translational modifications, they need to be assessed to ensure that there is correct amino acid sequence with the required biological function. And also pegfilgrastim polyethylene glycol attachment must be consistent as it affects the half-life, stability, and immunogenicity. Oxidation, deamination, and other primary structure in the post-translational modifications can impact the stability, efficacy, and immunogenicity. So the higher life, the higher order structure and aggregation, through that we'll be assessing the proper secondary and tertiary structures of proteins formed during folding and conformation, which are critical in acquiring the required structure and shape of receptor binding and functions. Additionally, protein aggregation can trigger immunogenicity and reduce drug effectiveness. Analytic methods like size exclusion, chromatography, and dynamic light scattering are used to assess this. Also, biological, there is also need to determine the effective. There is need to determine how effectively the drug interacts with the granulocyte colon stimulating factor receptors. There's also need to measure the ability of both pegfilgrastim and filgrastim to stimulate neutropenia proliferation and function, ensuring clinical safety. And lastly, there is also need to test for impurities that may arise during the manufacturing process, such as host cell proteins, DNA residues, and endotoxins. Degraded, truncated, or oxidized forms of the product, which could reduce efficacy or increase immunogenicity, also need to be assessed. So the next question states that how did ProMage perform in this test? So as I have highlighted before, that the primary structure, the post-translational modification, higher order structure, purity and impurity profiles in biological process, they proved to be similar between ProMage and Neulasta. So this table summarizes the physiochemical and biofunctional tests that were done to compare Neulasta and ProMage. So question 6 says that would you recommend the approval of this test? Based on the available data, we would recommend the approval of ProMage as a biosimilar of filgrastim. The comprehensive testing program demonstrated the similarity of ProMage to the reference product in terms of quality, safety, and efficacy. So I will hand over to my colleague, Ms. Trace, to continue.
Thank you. Good afternoon. I'll be taking you from question 7, which reads, what is your opinion on the comparative pharmacokinetic data? And to answer the question, ProMage demonstrated PK comparability to its reference product, Neulasta. ProMage and Neulasta, they were tested in a study with 172 healthy volunteers to see if their PK profiles were the same. In the pivotal study, it utilized a crossover design in healthy participants, ensuring a sensitive detection of differences between products, thereby reducing variability and making the study more efficient. The PK parameters, such as the maximum concentration and the area under the curve, they were core primary endpoints and they showed no clinically meaningful differences. And also the regulatory rule of bioequivalence, it says that the PK value should range between 80 to 125% of each other. And both ProMage and Neulasta, it made this requirement, proving that it is bioequivalent to Neulasta. I will move on to the next slide, which is question 8. In which indication was ProMage used as described in the European Public Assessment Report? The indication was ProMage was tested in the indication for the reduction of the duration of neutropenia and the instance of febrile neutropenia in patients treated with cytotoxic chemotherapy for malignancy. Question 9, what were the endpoints used in the human studies? And what is your opinion on the validity of these endpoints for drug approval? So the endpoints which were used were pharmacokinetic endpoints, there were pharmacodynamic endpoints, the immunogenicity, and the safety endpoints. And the endpoints, they were valid, they were clinically relevant, and scientifically sound. Looking at the package, what would be your recommendation? We recommend the approval of PERMEC for its indicated use. PERMEC, the comparative pharmacokinetic data, it demonstrated bioequivalence, and the clinical studies show that PERMEC is effective in reducing the duration of neutropenia and the incidence of febrile neutropenia. And we also recommend the ongoing post-market surveillance and more stability tests would be needed so as to confirm the long-term safety and also consistency. And if we're to request more data, we would also request data on the long-term stability data. We'll also request data on the use of the PERMEC in specific patient population, for example, in the elderly and in pediatric. Would also ask for monitoring product life cycle immunogenicity and risk management plans for ICSR reporting. How many patients were involved in studies, including in the submission package? So a total of 268 participants that were enrolled in the clinical program for PERMEC, with 172 in the pivotal study and then 96 in the supportive study. These studies, they were conducted in healthy volunteers and focused on PK and PD, immunogenicity and safety assessments. And do you think it's essential to require studies in patients before approving biological products? And what is the position of your regulatory agents? So, yes, there are cases in which it's essential. For example, when new molecular entities or there's a new mechanism of action, that is when a new drug is being assessed, and also when there's a significant difference in formulation or the manufacturing process, since this might affect how it works, and also when there are concerns about the immunogenicity or the safety of the drug, and this might reduce the effectiveness and result in severe side effects. And also there may be the cases where the patient trust can be waived, and this includes cases where there's a biosimilar with high similarity to the reference product, and the product with the established safety and efficacy profiles, and also when reliance module is used. I'm now handing over to Dr. Nyambayo to carry on with the presentation.
Okay, good afternoon, everyone. So thank you so much for this opportunity. So we're still going through a series of questions here. So the next question is, what is the current experience in Europe regarding the critical value of patient trials and biosimilar approval? Discuss supported by appropriate references. Is that opinion been adopted by your agency? So yes, MCAS, we've adopted the EU, US FDA, and the UK agents, MHRA, in prioritizing, and although they submit an application for registration in an ECTD format and we evaluate it. However, we also stand guided by the reliance model. Reliance doesn't mean that you just let things go, but you have to evaluate the dossier and make sure that everything is actually making sense. And we also review literature reviewers or published literature to see what is happening. So the PELMEG example was a very good example that in 2019, we actually registered it based on those two PKPD studies. That is like 6 years ago. And then what we also find is that there's quite a lot of comparative guidance document for each biosimilar. So you can also look at that. And also the advantage with us in Zimbabwe, we find that most innovator products, the companies apply for registration after they've been registered elsewhere. And when we talk of EU, it's about 26 countries. So then they are required to submit the periodic safety update reports, and that information is available online. And we also compare notes, and we also look at the EMA assessment reports in addition to our own assessment reports. So yeah, so there's really clear guidance on the stepwise approach, as my colleagues have already indicated. So the next question is, if you compare the 2008 ZASIO and 2018 PELMEG in the case of the EPARs, what do you see as the striking difference? And as you can see, the years, there's a 10-year gap. So obviously ZASIO went through a more rigorous approach because of the limited information that was available at the time, and we put the hyperlink and you can read it. It's quite interesting, the differences in the studies that were conducted. So there were two pivotal safety studies conducted for ZASIO, and then for PELMEG, it was the two PKPD studies. And then this table is just trying to show you the differences in terms of the clinical studies which were conducted. And also the reference product, that was quite interesting, that ZASIO reference product was the Neopagan. And for PELMEG, the reference product was Neulasta, because of obviously the biochemical and physiochemical similarities. And so, yeah, so that's really the major differences, and the data is really available in detail if you're interested in reading it. And then the other question is, what is your opinion of applying the principles of reliance in assessing the products in your agents? So we really agree with the international guidance for these new molecules. However, you find that when it comes to biologics like monoclonal antibodies and all these other products, find there are different guidance documents, and the requirement for MCAS is that they still have to apply and supply a full ECTD dossier submission, and then we evaluate. And then, like, for instance, monoclonal antibodies, there was an interesting publication done where an ethnicity requirement in the Japanese, Asia countries, South Korea, Japan, and you find each country has different requirements for ethnicity. But studies have shown that monoclonal antibodies, there isn't much difference. So the requirement might just be now country specific, but it's not really essential. So it's always important to know each product and the history. And also for emergency preparedness, when there's limited information, then we really have to join hands as regulators in the SADC region, the Zazibona model, and also consider the international regulators' guidance documents as well. So there was also a question on the UK MHRA that in 2021 they adopted as a policy that in most cases a comparative trial in patients may not be necessary. And also, we also looked at that. There were also some references from our lecturers. So the question is, in which cases do you think a patient study is essential? And when do you think a patient trial could be waived? So for biosimilars, if the reference product is quite a comparative and guidance document don't require the patient studies, then they can be waived. However, the pivotal PK/PD studies should be done. And we really liked the MHRA guidance document. And there was a statement there, and that's the link you can actually Google. We really enjoyed reading this guide because it says that, because I think the concern as well from clinical practice is the products interchangeable. If then you have got your new biosimilar, which has been compared with a reference product. So the conclusion from the UK guidance document was that once authorized, a biosimilar product is considered to be interchangeable with the reference product, which means a prescriber can choose the biosimilar medicine over the reference product or vice versa, and expect to achieve the same therapeutic effect. Likewise, a biosimilar product is considered to be interchangeable with another biosimilar to the same reference product. And so the guidance further explains that as a result of interchangeability, switching patients from one product to another, whether reference product or biosimilar, has become a clinical practice. And we thought that was really reassuring for the patients and for the healthcare professionals. So these are the reference guide. And for Pelmeg and Neulasta, there was a very good publication by Roth K. Ichter, 2020, that really went to town about all the values of the PKD studies and all that, confidence intervals, Tmax, this and that. And it was really a joy to read that. So there is a lot of information, and we thank you for the privilege to participate in this group work. Thank you. Over to you.
Yeah, thank you to the MCAS team also for that excellent presentation, which together with the previous one gave also a very good overview on the topic of this medicine, this biosimilar, also about the changes over the historical time, and specifically here also your agency's approach. So that was highly appreciated. We come to the end of this session here. I would like to ask Lise still if you have any urgent final comments on this presentation. Otherwise, I would suggest we close it here.
Thank you, Bernd, and also congratulations from me to this group. A very interesting presentation. Specifically, I appreciate the table, Zarzio, Pelmeg, the comparative table. Also, your mention about interchangeability, which I don't think any of the other groups touched upon. So that was, I think, also interesting for others to hear about. Maybe one last question, very short one, because I also want to give you the opportunity to answer to a question, is different from the other questions. We know that sometimes patients, healthcare professionals have doubts about biosimilars because it's a rather new concept. Why is it a similar product? Why is it not identical? They have questions and doubts if they can use it as safely and effectively as the reference product. What would be, as a regulator, your key messages to confide support in biosimilar use to such a concerned patient or healthcare professional?
Okay, so are you talking about advice to patients or advice to healthcare workers or both?
Yeah, indeed. What would you communicate to a patient or a healthcare professional on the safe and effective use of biosimilars? What would you like to share them about the science that underpins the evaluation and approval of biosimilars?
Okay, so our approach for such products as MCAS has been a reassuring approach. So as you know, in the EMA, it's a legal requirement to have the patient leaflet as well. So for such products, we also insist on the patient information leaflet. And for us to get the GPT maturity level 3, one of the indicators for market authorization was that we have to make that information publicly available. The patient information leaflet plus the summary of product characteristics. So then what we then do is, it's more, as you know, cancer is not an easy condition to manage. So normally the communication goes through the healthcare provider, through the patient. But obviously, because of the Google and availability of social media, the patients actually know more than the doctor these days when they go to see the doctor. So we really encourage that collaboration between the patient and the doctor. And as we say, the patient information leaflet, which we insist on, we find that the language is really simplified, that the patient can comprehend. Thank you. Did you hear me?
Thank you. And I may take the prerogative as a chair to say that closes the session now. And many thanks again. And certainly one of the important aspects
So
regarding
the reason why we emphasize on the patient information leaflet, approved patient information, because patients want reassurance that the regulators have really thoroughly looked at this. And so if it's an approved document which is going to the patients, we find that actually builds the trust. Thank you.
Thank you. Also important point. Yeah, and my last point would have been that actually also well-functioning regulatory agency provides a very good reassurance. And for that, I'm very glad to have so many enthusiastic young scientists in the regulatory agencies who have attended, and also outside, who have attended this course now here. That is great. And that is, first of all, many thanks to all the presenters during the day today. This, for me, was again a wonderful session with excellent presentations and discussions. Specific thanks to Lise Barbier for chairing the question sessions. And of course, also to all the other faculty members, to the organizing team. Thank you. With this, I would like to hand it over to Stephanie for some final organizational comments on the MCQs and anything else that we need, messages that we need to convey.
Thanks, Bert. Hi, everybody. I just want to quickly wrap up, so I'm not going to hold up too much of your time. I just wanted to remind you that in order to complete the course, there is still that outstanding component of the individual exam, the 2-hour exam, that is scheduled for next week. Please be encouraged to complete your self-study assignments on the Moodle platform as soon as possible, as these contribute 10% of your year mark. And we'll be completing all of the assessment criteria leading up to the exam during the next week. So please try and complete those tasks before Monday next week in order for those marks to reflect your effort. In terms of the exam, you can find the exam at your normal Moodle platform. This is hidden right now for you, but next week, Thursday, you will find the final exam underneath the group work assignment. When you click on it, it'll show you this link that you'll click on, the ClinReg2024 final exam. And just as an overview, you'll have 24 hours in which to start your exam on the 13th of February. But once you start it, you only have 2 hours in which to complete it. So if you don't complete your exam within that time frame, you won't be allowed to do it again. Just as a final reminder, the exam also only consists of 30 MCQ questions, and each question has one best answer to choose from. Again, the final exam contributes 50% of your year mark. But if you are having any concerns or technical difficulties during the time that you complete your exam, please feel free to reach out to me or alternatively to elearning@pmxafrica.org so that we can assist you. Lastly, once your exam is completed, we will be sending you post-course evaluation surveys to complete. Please don't ignore my emails. Please complete the immediate post-course evaluation as well as those that will be sent to you in 3, 6, and 12 months. Following completion. This is just for us to track how well you're doing in terms of implementing your skills that we've taught you over the last couple of months, and also how well you have attempted or achieved your objectives to the course. And then the most exciting part is that once you've completed your exam, we can head into the graduation ceremony. So more information regarding the graduation ceremony will be sent following the exam next week. So just keep an eye out for any communication coming from our office. And with that, I wish you all the best. And thank you so much for an incredible cohort this year. We wish you all the best with your exam. And yeah, thank you so much for today. And thank you for everything. Thanks, Brent.
Thank you. Bye-bye to all, and looking forward to seeing you at the successful graduation ceremony.
Thank you. Bye, everyone.
Bye.